I hope you found my previous talk interesting. I'm going to be drilling a little more now into this part about multiple mating, which has been my chief research interest for about the last 7 years. I think my title kind of says my overarching philosophy about it. Multiple mating, the overlooked key to bee breeding. So I hope I can make that point and that argument clear, and hopefully help effect some change in our breeding practices as a result of this research. So here we go. Okay, what we remember from our last lecture, in a social insect nest, we have divided duties. We have this strong division of the females in the social honeybees. We have the female who reproduces, and then we have the females that do all the rest. A strong division between colony work and colony reproduction. Now, if you remember too, that going back to your high school or college biology, you remember Darwin's great idea. And his great idea was that organisms evolve over time because our habitats are continuously testing our genomes. And as long as our genomes have the characters that we need to survive in that habitat, then we continue to reproduce and propagate those genes. And the flip side of that, of course, is also true. Those individuals who are not fit in their habitats, by extension, they die. And so their genes are not propagated. So it's simply a statistical project, isn't it? That success breeds the accumulation of genes that are, by definition, fit for that habitat. But it's all predicated on this survival thing. You got to survive, you got to reproduce. Well, Darwin was keenly aware of the one big anomaly to his theory. He wrote about it at length in The Origin of Species. He used different language than you and I are accustomed to. He called them the neuters in bee and ant societies. He was fully aware that they not only do not reproduce, they are not capable of reproducing. And if that's not bad enough, many of them, including the worker honeybees, they will self-sacrifice if necessary to help someone else reproduce, in this case, their mother. And Darwin understood that, superficially at least, that this was a challenge to his theory, which was predicated on reproduction. If you don't reproduce, you are not playing Darwin's game. So this is another little interesting aside in the history of biology. Those of you who had high school or college biology, you'll remember Gregor Mendel. He was the monk who was a contemporary of Darwin's. And Gregor Mendel is the one who figured out the mathematics of inheritance. And he was using pea plants in his gardens. And he was looking at peas that were wrinkled versus peas that were smooth, and peas that had different flower colors than others. And just by pure trial and error, well, intelligent trial and error, he was able to figure out the mathematics of inheritance, that we use to this day. And you remember, in a nutshell, that, for example, many genes have dominant forms and recessive forms. And if you inherit a recessive from your father and a recessive from your mother, then you're going to express the recessive form of that gene. But if one of your parents, and it only takes one, if one of them has the dominant form of that gene and you inherit it, then you're only going to be expressing the dominant form of that gene. You can be carrying the recessive form and pass it along, but you're going to be expressing only the dominant form. Well, Gregor Mendel figured all this out, just using pure and simple mathematics and keeping track of pea plants in his garden. Now, one of the interesting facts about history is that Gregor Mendel and Charles Darwin were contemporaries. They lived there at the same time, but there is no record that they ever met or that they were ever aware of each other's work. If Darwin had been advantaged with the work and the knowledge of Gregor Mendel's laws of inheritance, he may have been able to crack this puzzle, but he didn't. And so he went to his grave essentially frustrated about the problem with the neuters, as he called them in the social insects, which seemed to defy his theory. And so that's the state of the problem for about 100 years, until the year 1964, when a young postdoc, Bill Hamilton, at University College in London, published this seminal paper in the Journal of Theoretical Biology called The Genetical Evolution of Social Behavior. And Bill Hamilton said, you know what? There is a possible explanation for altruistic, self-sacrificing, non-reproducing behavior. And he worked it all out mathematically and then showed how it applies to real live animals, including honeybees. And here's Hamilton's great formula. And it is a formula. It's a formula of math. And I'm going to show it to you because every one of you can understand it. And it may be something that comes up in a dinner table conversation someday, and you'll really impress your listeners. It's called Hamilton's rule. And here it is. Hamilton said in 1964 that altruism can evolve in a population if the following conditions are met. If the cost, that's the letter C, if the cost of the altruistic behavior is less than the benefit to the recipient times their shared relatedness. Altruism will evolve in a population if the cost to the altruist is less than the benefit to the recipient multiplied by their shared relatedness. Now, John Haldane, who is a contemporary of Hamilton, he put it this way, which I think makes it a lot easier. I would lay down my life for 2 brothers or 8 cousins. Now, he said this as a joke, but the math is right. If you add up the genes that you share in common with your brothers or even your cousins or even your nephews, if you can raise enough of those individuals, it will mathematically add up to the same amount of genes that you pass on if you rear your own child. It's pure and simple math. I, for instance, have 50% of my genes in common with my sister. My beloved sister, who lives in Logansport, Indiana, she has 50% of her genes in common with me. So biologically, using Hamilton's math, it is equally to my benefit to preserve and support and encourage and help the survival of my sister as it is for me to preserve and support and encourage the survival of my one daughter who lives in the Washington, D.C. area. You know, by Hamilton's rule, I am invested equally in both of those 2 individuals. So it intuitively makes sense. It's a genetic, mathematical explanation for a phenomenon that we're all familiar with, and that is familial love and affection. Well, I want to talk a little bit about that letter R. What is relatedness? Pure and simple, it is the fraction of alleles that you share in common with another individual. Okay, the fraction of your genes, the forms of the genes, the alleles that you share in common with another individual. So it ranges from 0 to 1. You know, it's a continuum along that span. Well, let's think about this for a minute. I think it's worth lingering because it'll help you understand where I'm going with this conversation. Now, I already said I share 50% of my genes with my sister, right? And if you have a sibling, this formula applies to you as well. It looks like this. Both of you get half of your genes from your common mother, right? But you only get half of your common mother's genes. And this is because in the ovaries of every female and the testes of every male, there's this process called crossing over, where when the gametes are forming, they line up temporarily and they exchange allelic information across the chromosomes. And then the chromosomes separate and each of them form their respective sperm or egg. But this mixing up process in the formation of the gametes is called crossing over. And it is one way in biological evolution that nature has assured a large recombination and resorting of genes every generation. Recombination. Okay, so half of your genes from our mother, but only half of hers, plus half of our genes from our common father, but only half of his, because of the same thing. You know, genetic recombination and the formation of sperm. So you add up 0.25 and 0.25 and you got, there you have it, 50% of your genes that you share in common with your sibling. Okay, so far so good. Now let's think about the order Hymenoptera, which is the ants, drinking from that spilled sugar spot up there on the top left, the ants and the wasps and the bees. This entire order is distinctive because the males have only one set of chromosomes. Now they are haploid. This is a feature common across all of the Hymenoptera. It's a feature also common to varroa mites. So it's not just unique to the order Hymenoptera. It does happen in other arthropods and in other groups in nature. But that's important, and it has big implications for this formula. Let's do it again. Let's imagine that I am two workers in a honeybee nest. Okay, I'm two daughters, and we share the same father and the same mother. Well, what's that formula look like now? Here we go. Just like before, I get half of my genes from our common mother times half of her genes plus half from our shared father, but all of his genes, which adds up to 0.75 of our genes in common. How in the world did that happen? Because the Hymenopteran male has only one set of chromosomes, there is no crossing over in the formation of his sperm. Tom Renderer was on my committee when I was a grad student at LSU, and he called drones sperm with wings. And he's essentially right. Everything a drone has in his genome is in his sperm, 100% of it. There is no crossing over. So the result in Hymenoptera for two sisters who share the same father, they are not just sisters, they are super sisters. And that is the term universally for females in the Hymenoptera who share the same father. Now think about this and think back to my previous lecture. If I am a solitary bee, I'm a solitary female, and I mate with one male, then that means all of my daughters are going to be these kind of creatures. They're all going to be super sisters. And so if I am one of these daughters, I am faced with a very important decision. I can either fly out and mate and make my own nest and raise my own daughters and produce daughters with whom I have only 50% of my genes in common, or I can stay home at the nest, help my mother produce more super sisters, and pass on 75% of my genes in common. This is pretty much a Darwinian no-brainer. And this was Hamilton's big revelation. This was Hamilton's big idea. He was able to show a very compelling genetic explanation for altruistic behavior, which would compel daughters to stay at their nest and help their mother produce more super sisters. I mean, what is there to not like about this? Sign me up. I mean, this is like hitting it out of the park. You know, for this, I can just stay home, you know, help mom a little bit and pass on 75% of my genes. The deal. That's what I'm going to do. And that, in fact, is the situation for the vast majority of species on Earth today. They live this solitary life. They live together in patches that sometimes, because of their activity, they create the false appearance of a social nest when in fact they are not. You know, each individual burrow is its own little individual thing, each mother doing her own thing and raising her own little brood. And so it does raise the question, well, why aren't they all social? Why not? What is there to not like? That's a great question. And a lot of genetic theorists have puzzled over that work. Ultimately, though, we find that the majority of species do, for whatever reason, choose to go the Lone Ranger route. There's something compelling, I guess, about my personal genes being propagated, even if it's only 50% of them. So just because we can do the math does not compel organisms to do what the math predicts, does it? Making that leap to staying at home and sacrificing my interests for the benefit of others remains a tough sell in the history of biology, and it's only happened 26 different times across all branches of taxonomy. We have social behavior showing up, of course, in the Hymenoptera, the ants and wasps and bees, but we also have it in the termites. We also have it in shrimp. There are species of shrimp that are social and live together in groups like this, where there are neuter shrimp that stay to help their mother make more shrimps. Probably the most interesting example is a rodent that we have in Africa called the naked mole rat. It's the ugliest thing that's ever evolved. It looks like a sausage with short little stubby legs, and it has no eyes. It's an ugly thing. But these rodents live together in burrows subterranean in Eastern Africa. They're counted among those 26 times that it's happened. But yet those species that have done that have had ecological preeminence. They have over-dominated their communities where they have evolved. So you have to be nuanced in the way we think about evolution. Is it common? No. In fact, it's quite rare. It's happened very few times. That thing about pooling together and sacrificing my own selfish reproduction is still a hard sell. But once those species have done that, they have thrived and flourished and filled virtually every niche on planet Earth. Interesting. A little bit of nuance there for us to chew on. Well, these simple solitary bees, as we're seeing here, tend to be monogamous. Single mating tends to be the rule. And this is a solitary, very common one that we have here in North Georgia. And you will find these matings happening on the ground. They don't do it in the air like the honeybees do. They do it on the ground. And because the females are mating with only one male, her colony, if she has a colony, are all super sisters. And for some species that do live together in colonies, in the Hamiltonian sense, their mother is mated to only one male. These do occur. They do occur. They're relatively rare. One big group that is heavily represented by these simple colonies is the sweat bees. That large family of bees. A lot of them are colored green, kind of a green metallic bee, and they're attracted to human sweat. A large fraction of those species live in these simple little colonies where there's one monogamous mother with a handful of daughters who live at the nest. And they are properly colonies, but they're not advanced colonies. At the end of the season, they die out. The workers, the daughters, it's not really proper to call them workers because they've not evolved into castes. At any time, a daughter could jump ship and say, hey, enough of this. I think I will go out and mate and try to make it on my own. And sometimes they do, but a lot of times they don't. And at the end of the season comes and they all just die. And the only ones that survive are the ones that got mated and they overwinter up. So it's a very committal form of sociology. They're kind of together for part of this, for the active season, but then they disperse at the end and they're not fully committed. And again, this is kind of what their little simple burrows look like. But for those species that did follow that simple eusocial path, inevitably specialization kicked in. We had some that were just genetically better at doing work and others that were genetically better at reproducing. And incidentally, specialization is not a product of evolution. You can take any two solitary individuals of a solitary species, put them together in a lab culture, and you will get spontaneous and instantaneous specialization. I promise you, one of them will take on reproductive tasks and the other one will take on nesting tasks. There's something, I guess, hardwired in the Hymenoptera to spontaneously specialize when they are forced to live together in groups. This should not be so difficult for us to think as humans because we see this happen all the time in human societies. Beekeepers clubs. You get a bunch of people together and you instantly see specialization emerge. Some people are Indians, some people are chiefs, right? Some take charge, some organize, some just like to set up the chairs. Others like to be the treasurer and take care of the finances. Others like to do public outreach. It happens, and that's just a product of the innate natural variance that already exists in individuals. So keep that on mind and chew on it. Specialization itself is not a product of evolution. It's spontaneously emergent from the principle of putting individuals together. Now, specialization can be elaborated by evolution, and it is elaborated by evolution in the case of the committed social species like the honeybee. So for these specializations' sake, we do see individuals that just get better at doing certain things, so that in time they did diverge into morphological forms like workers who are good for work and queens that are good for laying eggs. And once that morphological breach occurred, then and only then could we have multiple mating of the kind that I've been talking about. Now, why do I say then and only then? Because this is a patently bad deal for the daughters. Okay, up until this moment, they have been helping mom produce super sisters with whom they share 75% of their genes in common, which is a great deal, right? But once mom starts practicing polyandry and starts mating with large numbers of males and starts storing that sperm and producing a cohort of daughters that are no longer super sisters, the average relatedness in the nest drops to about 30%. That's a big hit. But why do they do this? How can they do this? How can this happen? This is great betrayal, right? Yeah, you're right. It is. It's big betrayal. So the mother's multiple mating is really bad news for the daughters. But it's great news for the colony, for reasons I had elaborated on in the previous lecture. It's only once you have a large degree of genetic variation under one roof that we start getting greater and greater specialization, larger and larger resource acquisition, which leads to larger and larger populations. You get that commitment to 12 months of living together as a group, and elaboration and size takes off and gives us the modern honeybee. I guess to put it simply, monogamous colonies and families are quite boring when it gets right down to it. It's only in the multiple mating colonies that we get the plethora of complex behaviors that we see in the modern honeybee. That spermatheca represents a gold mine, a trove of genetic resources that gives genetic diversity and variation to the colony. And it has been shown repeatedly that polyandry is positively associated with colony strength, health, and fitness. Well, why is it associated with strength and fitness? There's been some theories put forward to explain it. Okay, I get it. Right, you got genetic variation. Well, give me a little more than that. What is so great about genetic variation? Well, there's one idea that it reduces sex ratio conflict. Think about this. Go back to that early primitive Hamiltonian monogamous little bee family that I was talking about. If I am a daughter and I have 75% of my genes in common with my sister, then by definition, I only have 25% of my genes in common with my brothers. So if I look at mom's progeny, and from mom's point of view, she's happy to produce 50-50. You know, with every egg she produces, whether it's a male or a female, mom is passing on 50% of her genes. But her daughters that have chosen to stick around the nest and help make mom make super sisters, you know, I don't really like those brothers. You know, those brothers are a bad deal. I want to spend all my time making sisters. So it has been shown theoretically and shown in field that super sisters have a 3-to-1 preference for raising super sisters over brothers. And so in this incipient colony, there is an essential conflict in reproductive interests. The queen wants to produce 50-50, males, females. Her daughters want to produce more sisters at a ratio of 3-to-1. So there's a potential social conflict set up. So one explanation says that the mother just started practicing polyandry to diffuse this tension. By lowering the stakes down to where that's only 30 versus 25% in common, it minimizes the stakes for daughters to exercise their prerogative in raising other daughters. it lowers that sex ratio conflict from 75 to 25 to 30 to 25. Okay, that's one explanation. It's also been suggested that by practicing multiple mating that the queen increases the alleles in her colony, which reduces inbreeding at the sex locus. And I'm sure many of you, especially those of you who are interested in bee breeding, are well aware that one of the big effects of inbreeding in a pollination is that you get inbreeding at the sex locus. And when you get inbreeding at the sex locus, you get diploid males. And when you get diploid males, the workers eat them, so that they constitute a resource drain on the colony and they do the colony no good. And it's also a sign of inbreeding if you're a queen breeder. So if the mother mates with a lot of males, it reduces this problem. At an ecological level, it's been suggested that multiple mating and genetic diversity minimizes phenotypic extremes in the habitat. A lot of times evolution likes stasis. Okay, we like to glorify it and see it as this unbroken march forward for progress, when in fact sometimes the best progress is to just stay in the middle. You know, if you're an organism and you live in a particular habitat, you don't want to be at one extreme or the other. You don't want to be a sunflower that has the very super tallest flower, because you're going to get lopped off more easily, right? And the same token, you don't want to be the shortest sunflower in the field either, do you? Because you're going to be shaded out and not going to grow. So if you're a flower, you kind of want to be right in the middle with everyone else, right? Well, genetic variation minimizes the possibility of extremes being expressed. Genetic variation, there on the fourth bullet, may increase robustness against parasites and pathogens. If I have more genes in my toolkit, then I'm more likely to have whatever tool I need when the next parasite shows up. Okay, makes sense to me. And finally, it may improve task efficiency through specialization. A lot of the specializations that workers go through as they age are parsed out by subfamily, which father they come from. So even though they may go through all of the successions like a normal worker does, they may linger for a longer time in one particular group. So let's say if I'm an undertaker specialist, and yeah, it comes through my age-based period of time to be an undertaker, well, I may jump ahead and get to that stage quicker, and I may linger in there longer if I am a undertaker specialist, if I came from a father who carried that gene. So if I have a lot of fathers in the colony, if I have a lot of these possibilities for specialization, that all of this could add up to benefit for the colony. Okay, so far so good. This makes sense to me. You know, genetic variation is probably a good thing. But all of these are basically one form of the very same hypothesis, aren't they? Just the more genes, the better. So let's take a look at that with a little more criticism. Any of you who are agronomists, or any of you who are economists, are well aware of the laws of diminishing returns. If you are a corn farmer, for instance, you know very well that you can add nitrogen to your field and you'll get a yield increase, very strong at first. But after a while, that nitrogen is going to plateau, and you're just wasting time and money to add any more nitrogen because the crop will not make any more yield. This is the law of diminishing returns. The law of diminishing returns is very active when we are talking about the increase in genetic diversity, the number of alleles that a queen can capture as she multiply mates. Well, here's the numbers. Take a look at this. After the 6th mating, a honeybee queen has already captured 90% of the alleles in her breeding population. Like, whoa, after only 6. Well, that's interesting, but let's add to that the fact that in the genus Apis, they routinely double that. The species average, according to work by Dave Tarpey, he published back in 2004, Dave did a whole review of the entire genus of Apis, and he showed that for Apis mellifera, our honeybee that we raise, the average number of matings is 12, which is exactly double that asymptote. Well, you got to ask yourself, what are they gaining? Well, it gets worse. There's records in the literature of mating numbers as high as 44, and the highest, to my knowledge, is again by Dave Tarpey, but about 20 years later, one of his students found a naturally mated queen with 77 males. Now, obviously, she is literally off the charts. But you gotta ask yourself, why do they keep going? Look at that. If they've already achieved 90% of the alleles in their population after the 6th mating, why in the world should she keep on mating? Mating is risky. There's birds out there, there's predators, there's bad weather, there's sexually transmitted diseases. There's all sorts of reasons to minimize the number of mates, but yet they do. In fact, at an average, they exactly double it. They could get by with 6, according to the genetic variance model, but routinely go up to 12. What explanation for this? Well, one explanation could be that it's just purely chance. And when you think about those chaotic mating bouts of the queens, they are chaotic. You know, the queen's up there in the drone congregation area. The drones are fighting for opportunity to mate with her, and she may have little or no control over the outcome. You know, there may be 20 guys that just happen to be in the right spot at the exact right time, and it's like pow, 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 pow. And she mates with all of them within a matter of seconds. And the next time she goes out, you know, there's nobody else out whatsoever. It could be a perfectly random event. And Dave Tarpey has actually gone on record, you know, proposing that as one option, that it's not evolved whatsoever. It's a product of chance. Well, I don't believe that. I don't think that is true. I think there are adaptive reasons to go beyond that asymptote of 6 or 12 matings. The reason I know this is for a paper that I did in back in 2015 when I was on sabbatical in England. And we used instrumental insemination to create 3 different classes of polyandrous queens. We mated some with 15 drones or 30 or 60, which was bracketing a natural range of matings. You know, 15 is pretty close to Dave Tarpey's average of 12. And then we doubled it, and then we doubled it again. And we were able to show that as we jumped from 15 to 30 or 60, we had more brood in the colonies that were more polyandrous. And what really excited me was the graph on the right. As we jumped from colonies inseminated with 15 drones to 30 or 60, we had a significant drop in the varroa mite numbers. More brood, fewer mites, at polyandry levels that are wildly in excess of that flatlined asymptote that the genetic variance model suggests. What could explain these additional benefits at such higher ranges where the marginal gains in genetic alleles is just so small? What's going on here? I think the best answer to this came out in a paper in 1998, which I think has not received the attention that it deserves. And this is a paper by Stefan Fuchs and Robin Moritz in Germany. And they said, you know what? We have a theory, we have an hypothesis, what we call the rare allele model. And they proposed in this paper that extreme levels of polyandry could in fact evolve in a colony for traits that were highly beneficial only when they were rare. Only when they were rare. Well, you have to wonder about that. What kind of a gene could be really, really good to have, but only when it's rare? Well, it could be a gene that is so potent at one extremely important thing that it renders the colony incompetent at everything else. Imagine a colony that was so good at hygienic behavior that it jettisons all of its brood. Okay, that's not theoretical. It's actually happened. Marla Spivak, who's done her work on hygienic behavior, who's made her name in that field, has been able to manipulate colonies so that they have so much hygienic behavior that they kind of go crazy and they identify all brood as failed and contaminated, and they jettison all of it. Too much of a good thing, right? So it is not implausible, the condition that Fuchs and Moritz put forth. There may be some genes out there, it's kind of like Tabasco sauce, right? You want a little bit, but you don't want to eat a whole plate of the stuff. You want those specialized genes for that one really rare particular thing that that colony might confront in its habitat that it needs help for. But if all the workers were that one thing, then they wouldn't be able to forage, or they wouldn't be able to thermoregulate or defend their nest or anything else that's far more fundamental. So here we have a sixth bullet to explain evolution of high levels of polyandry, that it increases acquisition of rare specialist alleles that are only beneficial when they occur rarely in the colony. Well, what's a good example of that? Well, I would propose to you hygienic behavior. We can replicate any of these conditions through the use of instrumental insemination, as you see a device here. So what we did, my colleagues, Debbie Delaphane at the University of Delaware and myself here this past year, this is a paper we're getting written up right now, we were interested in trying to test this hypothesis, and while testing it, maybe find some information that's useful for the beekeeping industry as well. Kind of kill two birds with one stone. Answer an evolutionary puzzle and improve beekeeping at the same time. And we zeroed in on hygienic behavior because to us, it seems like an ideal candidate of the kind of rare specialist allele that Fuchs and Moritz were talking about. And what you're seeing here is a diagram of how one can select for hygienic behavior. At the time, we were using rolled sheets of just tin sheeting, and nowadays we use PVC pipes. But you can kind of screw a PVC pipe down into sealed brood, pour in liquid nitrogen, which kills the brood, freeze kills it, and then you can put that killed circle of brood back into colonies and monitor those colonies, their ability to remove that freeze-killed brood. The colony on the bottom right has done a marginal job at removing the freeze-killed brood, whereas the colony on the left has done a 100% job of removing the freeze-killed brood. So if you are a hygienic breeder, you would want to graft from the queen on the bottom left, not the one from the bottom right. It just so happens that hygienic behavior is also recessive. They are controlled by only about 7 genes, 7 loci, and they're all recessive. It only expresses when it's recessive. So we were interested in combining these two principles, polyandry versus classical genetic selection with a rare specialist allele, hygienic behavior, against varroa mites. And with instrumental insemination, we could create this experiment. And here's what it looks like diagrammatically. We created 4 classes of colonies with 2 levels of polyandry, low polyandry, high polyandry, and 2 levels of rare alleles, either pre-selected for varroa-sensitive hygiene or non-selected drones of a non-selected wild type. These wild type drones, let me stress, were selected for nothing, no selection whatsoever. They were just drones that we, colonies that we picked out of our apiaries. Now look at the polyandry column, and you can see that our levels of polyandry differed between Delaware and Georgia. In Delaware, they had large overwintering loss of their drone source colonies, so we had to modify our polyandry design. In Delaware, our low polyandry was queens inseminated with 1 male, and the high was queens inseminated with 4, which is admittedly on the low end. But in Georgia, our overwintering survival of drone colonies was quite good, so our level of low polyandry was 9, and the high level was 6 times that, 54 drones. And with using instrumental insemination, we were able to, with a great deal of precision, create these 4 different classes of colonies. All of the queens used in this experiment were all super sister virgins. So we tried to minimize maternal variation so that we could really zero in on the drone semen, which contained the rare alleles in the case of the VSH, or also control the drone dose, either low or high in our respective states. Debbie Delaney's crew came up from Delaware to join mine, and we had a big day of it in April of 2018 when we were setting up. That's Kirsten Trainer, by the way. They're on the far left. person for a time was the editor of the American Bee Journal, and she's now launching her own bee magazine as well. And there's the rest of my crew. And there we are, UGA, we are taking advantage of our research on using circle apiaries to minimize drift and try to minimize spurious variation in our study. And this is what our apiaries looked like. And then we tracked each one of these colonies with those different combinations of VSH versus polyandry to try to see which effect was the most important or if they interacted with each other. So I'm going to show you some colony strength measures after we measured these colonies. On the left, we have Delaware colonies. Let's take a look at brood and take a look at comb. In Delaware, polyandry significantly improved both brood production and comb construction across the board, irregardless of whether it had VSH or not. These are the overall averages looking just at polyandry. And I think this is worth pausing to think about. Think about this. If you are a queen breeder, and if you have gone to the bother of selecting for some trait and propagating that trait, you know how hard queen breeding is. It's just not a cakewalk, right? It's hard work to select for and propagate a particular line year after year. One of the things that I find that's repeatedly coming up with polyandry research is the benefits of polyandry itself, minus any genetic selection. Think about that. If you just simply pump the queens full of semen, you get fitness benefits, whether it's mite resistance, whether it's brood production, whether it's populations, whether it's honey production. And it sort of just jumps over all of that part about genetic selection, which is one of the biggest handicaps in classical bee breeding, is taking the time to do those measures. Polyandry seems to suggest you don't need it. You just need a lot of drones, a lot of genetic variation. So that is evident in this data set that I'm showing you. And this is one of many. It's similar to that data set that I showed you previous from my work in England. And it's been shown by many other authors. I mean, lots of people besides me are working in this. And it's a very fruitful area of research. And it is over and over and over and over again showing the benefits of polyandrous queens. Sometimes I think we can just dismiss the whole process of selection and just go for polyandry. That argument, I think, is not untenable. Okay, right. Take a look at the data on the right for Georgia. Once again, looking at the overall averages of polyandry only, we had a significant increase in the bee population and a significant increase in comb construction just in polyandrous groups as you go from low polyandry to high. So polyandry rocks, folks. I mean, take that message home with you. It's consistent. It's been shown over and over again in measures that are good for fitness and good for beekeeping. When it came to VSH, now let's look at the overarching averages based only on Varroa sensitive hygiene. There was only one character across the entire experiment in both states where VSH had an effect. It was significantly lower, ironically, for comb construction in Delaware. Yeah, okay, well, I do not have a well-reasoned explanation for this result, but I can offer one idea for us to chew on. And this is true of any classical selection project. You cannot avoid this fact. And that is, any artificial selection scheme is a genetic bottleneck. When you select for A, you inadvertently select against B. And you may not know what B is. It could be something totally off your radar as a finite, limited human being. As you're selecting for something, you are selecting against something else, which may be as or more important than the character you're selecting for. This is not unique to bee breeding. This is true of all animal husbandry. We humans in our finite lifespans cannot know every contingency. This, by the way, is the big advantage of natural selection. Natural selection does have the time to deal with every contingency. We don't. So our breeding programs will always be inferior to natural breeding programs. Our breeding programs are always genetic bottlenecks. So I can only say that this result right here suggests that in selecting for VSH, we may have lost something. You know, maybe comb, I don't know. But this data like this exist in other papers too. So I do think this may be something for us to consider as breeders. Okay, let's get now to what I think is really interesting, and that's the mite situation. And remember our hypothesis about the rare allele model. In Delaware, what you're looking at here is the mite counts. We're looking at polyandry levels. Let's just focus on Delaware. What you see here is a big parallel effect, right? There tended to be an increase in mites as we moved from 1 to 4 polyandry. There was just an increase there, and the effect was totally blind to VSH. Whether it was VSH or whether it was wild type, the trend was the same. This is what we call an absence of an interaction. VSH and wild were behaving identically the same. They were each responding only to polyandry. But you see that effect is non-significant. See that? So at low levels, that p-value is only 0.865, which is not significant. So at these low levels of polyandry, using VSH versus wild, we found no benefit for mite control. Take a look at Georgia. In Georgia, we had a significant interaction. We found that mite control, the mite benefit of VSH alleles, only expressed at the high polyandry colonies. VSH only expressed in those queens that were inseminated with 54 VSH drones. This is consistent with Fuchs and Moritz's model, that high levels of polyandry evolve and are responsive to gene sets that are highly specialized for some particular fit function, but only effective when they are rare. And this suggests that model is accurate for the evolution of polyandry at high levels. Let me say it again, VSH only expressed at the high levels of polyandry in Georgia. And it was a significant decrease in those queens inseminated with 54 VSH males. This is a good thing for beekeeping. This is what we call a good synergy. When you have pre-selected alleles that exist, they're not theoretical. We can go out and find them. We can use them. We can inseminate our queens with them. And we show that we get exaggerated benefit in those queens that are inseminated with large numbers of males. Let's take a look at survival. What you're seeing here, the red area is the numbers of colonies that survived. Okay, this is an either or kind of data set. They're either dead or they're alive. The blue is the dead colonies, the red one is the alive colonies. And here we're looking only at Delaware. In Delaware, colonies significantly survived better if they were wild type. Okay, this once again kind of goes back to that genetic bottleneck scenario that I'm suggesting. VSH may, you may lose something. You may be losing something of more general applicability and fitness with VSH. For whatever reason in Delaware, the wild types were significantly better survival. If you look at polyandry, you had significantly improved survival when they went from 1 to 4. So even within the range of 1 to 4, there was a benefit in Delaware, even though they tanked on their drone colonies and we had to truncate the size of the experiment. Even within that narrow range of 1 to 4, we got a significant improvement in colony survivorship with queens inseminated with 4 males. And then when you look at all 4 categories pooled together on the bottom chart, we see a stair-step increase in survivorship for the wild type stock inseminated with 4 drones. So if there was a benefit of polyandry, it was not apparent in Delaware, where the polyandry range was only a max of 4. I think that's significant. Take a look at Georgia. Things are quite a bit different. In Georgia, we did have a significant improvement in survivalship with the VSH stock. And jump down below to look at the reason why. The reason why is that oversized effect from the group that was inseminated with VSH semen and 54 drones. That was big enough to carry the day for the VSH colonies altogether. So VSH in combination with polyandry really worked in our Georgia replicate. I find this stuff extremely exciting because I think it's not only answering basic questions of biology, but it's giving beekeepers real and tangible stuff that is practical. So let's just kind of take a look at this. In this paper, in the two papers I've shown you here this afternoon, and in many others, trust me, this is a growing literature. Increasing polyandry is positively associated with colony fitness. Okay, it's just there's no controversy on that point. This is something that we as an industry need to start taking serious attention to and finding ways to deliver that in practical, usable ways. Remember that flat line that I showed you, that a pure and simple genetic variance model runs out of explanatory power after the 6th mating. However, honeybees routinely exceed that asymptote at least by double, very often more. So it's more than just simply, more genes are better. Give me more alleles. Once you start getting past that minimum of about 6 to 12, the reason she keeps on flying is so she can get the brass rings. Okay, she's already gotten the basic stuff taken care of. Okay, now what about VSH? Maybe one of these guys out there in the cloud is VSH. Okay, I'm going to go keep on mating to see if I can get with him. What about grooming behavior? Okay, maybe there's some ankle biter drones out there. I'm going to keep flying, so maybe I hook up with one of them. Or what about some kind of resistance character that we don't even have the pathogen yet to respond to? If I keep mating with a lot of males, then I'll be ready for that pathogen whenever it shows up. See, those are the brass rings, the icing on the cake. And the rare allele model is that icing. It's the extra stuff, the really good stuff. And you gotta keep going for it to get it. And evolution is selected for those alleles to be rare and recessive because they are so powerful that they could dominate the colony otherwise, and it would be no good at the more fundamental stuff. We find benefits of polyandry at incipient levels. Remember Delaware's survival? They got better survivorship when you jumped from 1 to 4. So in evolutionary time, these ancient mothers who started practicing promiscuity, which is what multiple mating is, they were very quickly able to deliver benefits to their colonies. Very quickly, they started getting fitness benefits from this genetic variation. But for rare traits, they had to practice more and more polyandry to achieve those rare specialist alleles. The bottom line being, a queen mates with males beyond a simple genetic variance asymptote in order to capture those rare beneficial specialisms, those brass rings, that icing on the cake. Now, this, in my opinion, is the best explanation for the wildly extravagant levels of multiple mating that we see in the honeybee today. I am grateful and indebted to many collaborators. I've been working a lot with Auburn University in the last few years, University of Delaware, and Emory, Barry Brosey, who's recently moved out of the Southeast up to Seattle. He's at the University of Washington now, a big loss for us. And of course, my crew there on the right, Jack Garrison, Jennifer Berry, Lewis Bartlett, and Catherine Hagan. All these individuals have contributed in some way to the research that I've shared with you today. And of course, our funders, which are largely from taxpayers like yourself, USDA, and the Foundation for Food and Agricultural Research, and the Georgia Beekeepers Association have chipped in a lot on this work as well. I might want to kind of give you a heads up. This team here at Auburn and Delaware and Georgia, we are in fact trying to find more simple ways to deliver the benefits of polyandry at industry scales. We have used instrumental insemination. And I still believe that AI, excuse me, I still think that AI is really the way the industry should go. It is the only way that we can absolutely control mating in the honeybee. I wish that instrumental insemination were more widely practiced. I had one of my master beekeepers point out to me that you can now download the plans for printing, 3D printers. Printing your own instrumental insemination device. So there you go. The costs keep coming down, down, down. And there are, of course, workshops available where you can go and learn this skill. Joe Latshaw in Ohio does this. Crispin Given at Purdue does this. Sue Kobe out in Washington does this. So these workshops are available, and the technology is going down, down, down. So that is one way. We are also studying whether we can use drone saturation in mating yards to try to increase the number of males available to virgin queens to mate with. Some people have suggested, and I think it's worth following up on, maybe this is something you can select for. Maybe you can select for promiscuity in queens. I don't know. Let's try it and see. So there are avenues that we can pursue to try to deliver this principle to the beekeeping industry. And with that, Larry, my formal comments are done. Maybe we have time for some questions.